Okay, thank you, Mr. Chairman. And first of all, I would like to express my sincerely thanks for the meeting committee for inviting and also for having a good opportunity to be here. My study will involve in the sperm physiology, and we were trying to preserve genetic potentials of the Estern Sarus cane, which we kept in the zoo. The first thing that I also need to acknowledge people as well. There are quite many people who are actually involved in this project, and I also need to thank for the financial support from the zoological park of the Thailand. I think all of you already know that the Sarus cane is basically divided into 3 subspecies. So the first one is the Indian Sarus cane, which is found in India, and also the Estern Sarus cane, which is found mostly in Southeast Asia, and also Australian Sarus cane. Unfortunately, the population of the Estern Sarus cane is classified as valuable in the IUCN Red List. So the population of the 3 subspecies has been estimated to approximately 13,000 or even up to 15,000. But if you look at the number of the Estern Sarus cane, which I think is the total is something like about 2,000 clan, which is mostly found in the Cambodia. So mostly found in Cambodia, southern Laos, and also southern Vietnam. And there are some 500 of the clan which is found in Myanmar. But unfortunately, so this clan has been extirpated from the natural habitat of Thailand, and also from Malaysia and also Philippines. So the problem that why they has been lost from the wetland of Thailand has been proposed by the effect of the agricultural expansion, pollution, and also wetland loss. People from many parts of the conservation center, like in the zoological park in Thailand, and also some of the conservation center has started to propagate the number of these Estern Sarus canes by either natural or even artificial insemination, and they actually succeed in producing some of the Estern Sarus cane in the zoo. But however, there are just only less than 200 canes which are being kept in the zoo. So in 2007, so the people from the zoo also tried to start to reintroduce the Estern Sarus cane and try to bring back them to the natural habitat. So this is the project that they have been doing at the moment, and I think you may know about this project, and I think you might hear something about the result probably tomorrow. But I'm not going to talk about this project, but I'm working on the sub-project of this reintroduction project of the Estern Sarus cane. So what I'm doing is I try to preserve the semen quality, so by means of the cold storage and also by cryopreservation of the semen. So the main objectives are to examine the effect of cold storage on the semen quality, and also we try to improve the fishing ability of the Estern Sarus cane. And this is quite important because you may imagine that these Estern Sarus cane are usually kept in the zoo, and some of them cannot pair in the young age. So sometimes they, what I would like to say is something like that they cannot mate with the female. So the thing is that we need to collect the semen and try to keep the semen even for the short term and even long term. So we conduct our research in the Nakhon Ratchasima Zoo. So we collect the semen from the 5 hen which they individually kept in the zoo, and we collect the semen approximately 2 times a month from April to August in this year. So after we collect the semen, so we divide the semen into the cold storage semen, which in the experimental 1 or for the cryopreservation in experimental 2. So in experimental 1, so once we collect the semen, so we dilute the semen with chem semen extender, which is from here, and then we slowly cool down the semen to 4 degrees. And then we look at the sperm quality at 24, 48, and 72 hours. And we use the semen which we collect immediately, and then we examine the semen quality as a control. And we use we look at the viability, and buoyocin, the viable sperm here. You can see that they have no colors, and this is that sperm with red colors. And for the experimental 2, so we try to cryopreserve, and we also try to improve our freezing techniques. So the first thing that we collect the semen, and then we dilute the semen with semen extenders, and then we try to equilibrate the semen with 2 techniques. The first one we call one-step equilibration, and the second one was the 2-step semen equilibration. So for first step, so we directly add the 7% dimethyl sulfoxide to the semen, and for the 2-step, we actually add a half of the cryoprotectant first, and then after we cool down for about 1 hour, we add another half to make the final concentration of 7% of DMSO. And after that, we load the semen into the straw and place the straw horizontally about 4 centimeters above the liquid nitrogen. And then after 10 minutes, we plunge the straw containing sperm into the liquid nitrogen for long-term storage. So after refreezing, so we store the semen using 2 different techniques. The first one, we directly plunge the straw into the 37-degree water bath, and the second technique, so we store the semen in air. And later on, we examine the quality of semen in terms of the sperm motility using the contrast microscopy, and also sperm viability using the fluorescent staining with a cyber protein. So the viable sperm will be green, and the Estern homodimer 1 with stain for the dead sperm, which you can see the red one is a dead sperm. And this is result showing you that so this is the time of the year that we collect the semen, and this is the volume of the semen. So you can see that it is a tendency that the trend produce more semen during June, July, and August. And the picture is much more clear if you look at the sperm concentration. So you can see that during April, May, the hen produces sperm quite a low number of the spermatozoa, and it's increased dramatically in June, July, and also in August. And the sperm motility and also sperm viability also seem to be quite high during the June, July, and August. And this is the result after we try to do the cold storage of the semen. If you look at this is a fresh semen, you can see that after cold storage, you can see the cold storage reduced significantly the quality of the semen in terms of motility and also viability. But however, our technique also can preserve the semen at least for 2 days, which we can obtain approximately 50% of motility and about 60 up to 70% for viability. And this is a result from freezing and thawing sperm. So what you can see is that we have really high quality of the fresh semen, but if we cryopreserve them, so the viability was significantly lower when you compare with the fresh sperm, and we didn't see any effect of the freezing technique or even the thawing technique on the sperm viability. But however, if we look at the motility, what we can see is that the picture is somewhat, I think, is approximately the same. That we can see that the quality of the frozen thaw semen was significantly lower than fresh semen, but we didn't see the. If we compare between the two equilibration techniques, so we didn't see difference. But if we look at the difference between thawing technique, it seems likely that thawing in air seems to be improve the quality of the cleansed semen in terms of motility. And this is the picture showing you that this is a fresh semen and this is a frozen thaw semen, so it's not working for this one. Okay. So this is just example what we have done in the laboratory since about 2 years ago. So from our data, so we can say that the semen production of Tharasanit is slightly influenced by the season, and also we also found some individual variation as well. And our results also tell us that we could preserve the genetic potentials of Estern Tharasanit by means of cold storage for at least 1 or 2 days. And this is quite important even if we have very low genetic diversity within the zoo. So basically we can preserve the semen, cold storage semen, and then we can exchange the genetic of the Tharasanit within the country or even within the zoo. And in this technique, so we don't need to transport any live animal, so it's safe for animal. And also the problem is the freezing and thawing actually influence and also markedly affect the sperm cryopreservability. But our technique, which I call field-friendly technique, can preserve the semen of the Estern Tharasanit, which approximately 50% viability and 40% motility. But the biggest problem is that the plasma membrane of the cleansed semen is really fragile, and I think it's quite easily to get damaged during freezing and thawing. So this might be that why when we try to thaw the semen in air, this is improve the semen quality about 5 to 10% when we compare with 37 Celsius thawing. And it might be that when we thaw in air, so the ice slowly melts and the water not really get into the sperm cell too rapidly, and this helps to protect the plasma membrane of the sperm cells. So in this case, so we found that the thawing in air is a good option for the cleansed semen. And the biggest issue is that we haven't done the fertility test of the fossil semen that we have in the liquid nitrogen. And this is quite important that once we cryopreserve the semen, we need to prove that the cryopreserved semen has or still remain the fertilizability. So we need to try to do the artificial insemination using fossil semen. Thank you for your attention.
Thank you. Any question for Dr. Theerawat? If there is none, I just got one question for you. Basically, your motility figures, your volume figures is based on 1 year result?
Actually, I did my experiment for 2 years, but I didn't include the semen characteristic last year. So I only present only this year.
Yeah, because it's just like an enrichment. If you first year you do it, your performance might not be good, but as you keep on going, the number, the volume will be increased. Semen physiology is my field, so I got problem there. Yeah, and the other one is thawing in air, because water is just like sharp edges when they become crystals. So they might poke your membrane, your plasma membrane. So it's contradictory to what I know. Yeah, so that area need to be researched.
Yeah.
Thank you.